So I want to switch gears, and we've just had a talk about standardizing the assembly of small snippets of DNA. I want to spend some time this afternoon talking about what I think is one of the more exciting things that's going to happen in the next couple of years, which is the standardization of the cells that we're putting these into. And some of you know I've been working for many years on simplified cells, and I wanted to give you a little bit of a perspective on some of the new thoughts in that area, but also to give you a little bit of an update of where we were. Synthetic biology is all about engineering things. It's an engineering technology, and that means we build things. And the things that we build, you'd like to have complete confidence that they really were the things that you were expecting them to be. You'd like to be able to model those. You'd like to be able to confidently go ahead and compare the results of an experiment with a model of the experiment that you'd done. So what does it take to get there? I wanted to remind people how much we've actually engineered E. coli already. And, so things started. We chose E. coli as a model system back in the 50s. We chose a non-pathogenic strain of E. coli. And that was an important move. We, and we would not be all as healthy, perhaps, had that not happened. Certainly my lab experience would, would indicate that. In the 80s, we transitioned from thinking about E. coli just as one of those bacteria that you grew to a cloning-friendly bacterial species, and we changed the strains of E. coli a fair amount during that process. We eliminated restriction systems. We made versions of the cells which did not have endonucleases, which were less prone to recombination. All of those things were things that happened. We developed expression-friendly strains a little bit later, strains that we could induce in a reliable way with high yields, and which were protease deficient. More recently, we've started seeing some additional strain development, things like the origami and Rosetta strains, which have better codon usage for mammalian and other species. Some weird things like the mutator strains for XL1 red. And very recently, we're starting to see George Church's and Joe Jacobson's and Pete Carr's work on E. coli, and the work at Scarab Genomics on removing insertion sequences and annoying features in the genomes. One of the things I'd like to plug here, Jason just finished talking about the problems that we see in assembly with the inability to select against accidental genomic fragments coming into the assembly process from impure plasmid preps. We think that the way to engineer around that from an assembly standpoint is to develop a special strain. The special strain will simply eliminate the fragments in the genome, eliminate the restriction sites that would cut those fragments in the genome. So we've had a long history of improving our E. coli strains, and and I think some of us take that for granted, but we shouldn't be doing that. If you go out into the industrial sector, of course, there's a lot of life other than E. coli, and so we have a lot of people working with Pseudomonas and Clostridium and even the yeast and Pichia world. Those people choose those strains for very good reasons. It's not just accidental that you go off and you start using Clostridium. It's that the cells have metabolic intermediates that may be critical to the system that you're engineering. They may fit into an environment that E. coli is not friendly to. They may be inherently safer. Some of the Lactococcus and Lactobacillus strains people are perfectly happy putting in food, and you wouldn't want to perhaps do that with E. coli. There may be issues with IP. Pichia, for example, is one that I think your typical industrialist would avoid like the plague because of the IP that surrounds that organism. So there's a lot of issues that come in, and there's life beyond. Even in the microbial world, there's a lot of life beyond E. coli. So I put forward as a strategy that I think, I suspect is not going to be complete in my lifetime, but a strategy that I think we are going to go towards. And Clyde Hutchison's talk on Friday touched on this matter with one of his last slides. The slide, I think he said, don't ask for this software. The software said basically some of the same things that are on this list. Think about designing a cell where you get the push button ability to say, what metabolic pathways do I want in this cell? What environmental niche do I want this cell to grow in? What kind of codon usage would I like it to have? What sort of membrane? What sort of biosafety control do I want in this cell? How do I want to be able to assure that nothing bad happens with this cell? And I think the way to go about that is to think about a very, very simple cell, and I'm going to tell you about my favorite simple cell, or some of you probably are bored hearing about it by this point. But to think about simplifying a very, very simple cell, already very simple cell, and to think about then adding back some required complexity. And instead of just thinking about individual genes and so forth, think about metabolic modules that you could swap back in. The organism I'm studying has no citric acid cycle. It just does glycolysis. Well, what would it take to add a citric acid cycle to this organism? And so let's think about that. We could think about modules that provide different energy sources. Arginine is not metabolized, but there are other mycoplasmas that metabolize arginine and use that as an energy source. Could we install photosynthesis? That actually sounds like it's perhaps very difficult, but might not be with some of the direct membrane inserted photoactive systems. We heard earlier today about the possibility of running the citric acid cycle backwards. Well, maybe we should be doing that. The organism I'm studying, the mycoplasma that I'm studying, has essentially no secondary metabolism worth speaking of. It can't synthesize amino acids or nucleotides or lipids or any of the cofactors. Those could be plug-and-play modular components. You could say, well, I'd really like an organism that can synthesize leucine. And what does it take to do that? I think that that's entirely plausible in a few years. So I got excited about this by reading a paper by Harold Morowitz, the second paper, The Completeness of Molecular Biology. There's actually an unpublished manuscript that had an even better title. It was A Complete Understanding of Life. Written in '84, and he pointed out that someday we were going to have a complete sequence for an organism. We were going to understand what all the proteins were and what they all did, perhaps, and wouldn't that be wonderful? And it's important, I think, to be able to concentrate on this point right here, the place where in our genome size where we transition between the things that are dead but interesting, and the things that are alive and extremely interesting. And we're right here. This is the organism I study, Mesoplasma florum. You heard on Friday a good deal about Mycoplasma genitalium. I won't try to repeat that. This is a picture of Mesoplasma florum. These are the colonies. They're a little bit bigger than the genitalium colonies. This is an E. coli cell in an optical microscope. This is a Mesoplasma florum cell. And this is a TEM of a Mesoplasma florum cell about 400 nanometers in diameter. So it's a really small cell. There's a false color image of the same image of the genome. We chose this organism because, as I sometimes tell people, absolutely no one cares about it. It's not a human or a plant or an animal pathogen. It's BSL-1. It won't grow at body temperature. It grows fast at 30 degrees. 40-minute doubling time. It's a facultative anaerobe. You don't even need to shake the flasks. It has a small genome and a small number of coding regions. So those are all features. This is the copy. This is a schematic of the genome. The red bars here indicate genes that are in the counterclockwise direction. The blue bars indicate genes that are in the clockwise direction. It's essentially all genes. The fact that you see them oriented in the direction of the replication fork indicates that this is a stable genome. It hasn't been infected by viruses and so forth. So a small number of tRNAs. In fact, there's a tRNA that's missing, and perhaps we can get back and talk about that later. There's one insertion sequence which is inactive. And as I said, the genes are oriented in the same directions as the replication forks. The codon usage is a little unusual. TGA codes for tryptophan, and CGG codes for is unused codon. It's not used in any of the protein coding regions in the organism, and the tRNA is missing, as I mentioned. We're busy trying to understand the metabolism of this cell, and the most recent project is a project to really build the flux balance analysis and the biopsych model. Some of you may be familiar with EcoPsych as a model of E. coli. We're doing essentially all of that again in this organism. And the key idea here is consider the elimination or addition of entire pathways in this organism. Here's a metabolic map. It's a little bit too detailed for you to see, but up on the top is glycolysis. In the middle here this is protein synthesis over here. This is nucleotide interchange. This is ribosomal construction down here. And so there's not a lot going on in this cell. It's hard over on the transporters. There's a great many transporters, some of which are uncharacterized, unfortunately, in terms of what they bring in and put out. The proteome has been done. We've identified 389 out of the 682 coding regions. This is a 2D gel with the spots marked that we picked for mass spec analysis, although we also did a whole cell version with MudPIT of the mass spec data. It's missing a cell wall. It's missing the citric acid cycle. No amino acid synthesis, no fatty acid synthesis. 1 sigma factor, about 10 DNA binding proteins for control. There's a good number of ribo switches. There's one restriction system, a SAU3A1-like restriction system, and some evidence, which bothers me, and I'm going to try to get rid of proteins that do more than one thing in the cell. So there's this general problem with when you try to simplify systems, if you try to reduce the number of parts too far, then each of the parts has to start doing more than one thing. And that's not a good thing from an engineering standpoint if your goal is flexibility and modularity. The most recent work we've been doing is to do transposome insertions into the genome. And this is what I'd like to spend a few minutes talking about this afternoon. The idea here is to try to identify the parts of the genome which are not essential. And the way we're doing this is with random insertions of transposons into the genome. The specific transposon we're using is TN5. we're using the epicenter transposon kit, which is a neat kit. The idea is the transposon inserts randomly into the genome and carries with it an antibiotic cassette. We did a recoded version of the TET-M gene, recoded for dual use in E. coli and in this organism. So we avoided all of the weird, or rather, coded around all of the weird codon usage issues. That's a BioBrick part, which you can go to the registry and find. It actually works better as a tetracycline resistance gene in E. coli than the standard ones. That antibiotic cassette, when it's inserted into the genome, conveys resistance to tetracycline, and also includes translational stops in all frames. After the insertion event, we grow the cells up on tetracycline plates. We pick individual colonies, put them into 96-well plates, grow up those plates, and then put the plate into the freezer so that we have a copy of the cells. So we have all of these in the freezer. And we do a Zymo genomic DNA prep, which this is not a paid advertisement. This is an unpaid advertisement. The Zymo genomic preps work really well, and they're cheap. And we sequence the transposon insertion locations directly from that genomic DNA prep. No PCR or anything else like that. Just a word about these transposomes. The idea is that you do an in vitro reaction between the DNA, the linear DNA fragment of the transposon, and the protein, again, in vitro protein, the TN5 transposase. And that reaction, when you finish it, the protein binds to 19 base pair inverted repeats at the two ends of the DNA fragment and forms this hairpin structure in vitro. That hairpin structure then is electroporated into cells in a fairly trivial reaction. We plate the cells out on a tetracycline plate, and the only things that grow on that plate will be the cells which have had a transposon inserted randomly into the genome and where that insertion event was not lethal. So those two things are both required. When we have picked about 2,700 of these, and they're in the freezer, most of those are insertions at fairly unsurprising places, a random lipoprotein or some protein with unknown function. There are some surprises at least to me. Some of you that study bacteria probably know about the FITZ gene, which is the central cell division protein. Not essential here. MREB, which is the major cytoskeleton protein, is also not essential. Many of the ribosomal proteins many of the ribosomal modification proteins, the ones that add methyl groups or sulfhydryl groups or decorate the ribosomal RNA in some way, those are not essential. Many of the tRNA modification enzymes are not essential. Some of them are, but many of them are not. And so this set of surprises it also includes some things that I still don't understand. The South3A1 restriction system appears to be essential. And if anyone has any ideas as to why that's true, I would like to hear that. I talked to Rich Roberts. He's never heard of anything like that either. So I'm looking for ideas. We're going to try knocking it out specifically. A lot of the GTPases that nobody knows what do are essential in this organism. NGA, OBGA. Someone please tell me what these do. We're busy comparing against the 3 other mycoplasma transposon insertion studies that are out there. 2 this year by Kevin Dybvig at Alabama, and the one that John Glass did a couple of years ago on Mycoplasma genitalium. The plan going forward with this organism is we're hot on the trail of a theta mode plasmid that will be functional. I'm quite confident that this recent work by Michael Breton in France, looking at the origin of replication for the PSCI2 plasmid coming out of Spiroplasma citri, will give us a stable plasmid in this organism. We're also pulling out the REC-T gene, also out of Spiroplasma citri, which is a homologue of the, it's a similar function to the lambda beta gene, which catalyzes recombination in the lambda red system. We're hoping that we'll be able to put that under a heat-inducible promoter and use that to help us edit the genome in place. So we're planning on doing homologous recombination to replace and clean up the genome in this organism. Much in the same way, there's a very nice poster on doing a similar thing in Bacillus out in the hall, if some of you may have an opportunity to take a look at that. We're bringing up a wiki annotation system, and P.J. Steiner is designing our microarrays for starting to look at what genes are present in what genes are not present in other organisms that are present in this one, by doing DNA array work, and of course the standard things of looking at transcription and small RNAs. I wanted to just very briefly touch on some work that I'm doing in collaboration with Ken Oy and the POET group at MIT, looking at what we can do in terms of actually engineering into organisms the ability to be confident about the biosafety issues. So one of the things that happens sort of automatically in this organism that is this codon isolation. I said that UGA is used as a tryptophan codon in this organism. What that means is, since UGA is a stop codon in almost the rest of the world, that means that when you transfer genes out of this organism, they're not functional typically. Similarly, if you have genes in the outside world which carry the CGG codon, and that's most genes, when they come into this organism, they're not functional because they can't be translated. I believe, of course I don't know, that this is the reason why the organism has a clean genome and no active insertion sites, insertion elements. Because when a virus, a phage, or a transposon tries to come into the organism, the CGG prevents translation. The lack of CGG codon tRNA prevents translation of those proteins, and basically nothing happens. So this is very interesting from the standpoint of biosafety, because now you have an organism where at least in some ways DNA or active genes can't transfer out of the organism, and also genes on the outside can't transfer into the organism. And I think we can enhance this with, it's a simple enough organism that we can go in and intentionally engineer this rather than just accepting this as a natural event, which is the case right now. We're looking at putting in a pair of required essential nutrients that don't occur much in the natural world. We haven't chosen those yet. If people have good ideas for them, I'd like to hear that. But the idea is, if you have a pair of essential nutrients, then it's very difficult to gradually evolve resistance to the lack of those nutrients. Whereas if you have a single one, then you can gradually evolve an ability to develop or use that nutrient or avoid that nutrient. Then the other idea is, again, to put in a pair, probably rather than one, kill switch, which uses a non-toxic, cheap, but typically not available in the environment chemical, which will allow us to just go out there and directly kill this organism if we need to in the environment. And again, I'm open to suggestions as to what such a chemical might be. We have some ideas, but maybe you just choose DDT or something. With that, I'd like to thank a couple of people, Greg Fournier, who helped me with the annotation originally, the Whitehead Institute, who did a lot of the sequencing work on this, Samantha Burke, who has been is an undergraduate working with me, who has done a lot of the transposon work, PJ Steiner, who's working with me right now on the array work, and our funders, and the MIT synthetic biology work. Get busy. Do we have questions? Yes.
You just mentioned that because of there are stop contents in there, so it could be make sure that it's biosafety.
It could be
biosafety, because there are stop content in the genes, so it cannot.
Yes.
But I found some research showed that this stop contents could also introduce unnatural amino acid.
Yes, that's certainly true. There's suppressor mutations in E. coli, and of course, it's the codon for selenocysteine in some cases, if there's the right structure.
Is it possible that these two combined together could cause some damage to the?
There's always that possibility, right? I mean, this is a complicated system. But the idea is to reduce the complexity of it to the place where we really are more confident rather than less confident of the safety. And will you ever be absolutely safe? Absolutely not.
Yeah, thank you.
You have to get over that as an idea.
Good.
I was wondering if you foresee an extension of this idea to like maybe a generic yeast organism or a generic mammalian cell. Again, sort of reduced complexity with modular things that you can add back in.
You haven't been looking at the posters.
Oh,
There's a poster out there on a group at Johns Hopkins that's is doing a rebuild of one of the yeast chromosomes with the intention of rebuilding them all.
okay.
So, yes.
Concerning the issue of modularity and the fact that proteins, as you said, they can do several things. Nature has invented membranes and compartments. Can you do anything with a synthetic compartment?
With synthetic compartment, well, these organisms, the specific organism I'm studying has two compartments. There's the inside and the outside. And there's not a lot of complexity there. But as you go up to higher organisms, absolutely. It starts to become very interesting.
With the mysterious SALV3A-like restriction enzyme, do you know if this is really active as a restriction enzyme?
Yes, I do.
It is, so it really is a restriction enzyme.
It really is a restriction enzyme, and you can't cut the genome at GATC.
Yeah, okay. Interesting.
Okay, thank you.